Okay, class, where we are now, we're looking at gastrointestinal 3, GI 3 in my PowerPoints, GI 3. All right, let's see what comes up to bat here. So we're talking about the pancreas. The pancreas, let's say, is mainly retroperitoneal, okay, behind the deep to the greater curvature of the stomach. There's the stomach. There's the greater curvature. Okay. It's behind that. It's behind, not in front of. That's why it's kind of highlighted. Head is encircled by the duodenum, tail or butts. So in other words, the pancreas has a head, a body, and a tail. A head, a body, and a tail. Let's look at that. So the pancreas has a head, a body, and a tail. Tail of pancreas, body, head of pancreas. Okay. So we go further. The endocrine function and the exocrine. Okay. So when we look at the pancreas, this is what we want to say. That the majority of the pancreas is an exocrine. Majority of the pancreas is exocrine. Okay. There's only little spots of endocrine. These endocrine spots are like little islands, like Galveston. They are surrounded by the exocrine. So the pancreas primarily is exocrine, delivering through a duct. Primarily exocrine. And see, the endocrine, these little islets that you see here, have alpha cells, beta cells, delta cells, F cells. Now, the beta cells are the ones that deliver insulin. So in other words, is not a lot of insulin cells in the pancreas. They're in these little islets. Most of the pancreas are in these acinar aspect, which is the exocrine part. And see, here's that exocrine. The digestive enzymes are made here, travel through the duct, and that duct adds, and then finally it comes here. Okay? So again, the pancreas is endocrine and exocrine, but about 99% exocrine, really, to be honest. Only about 1% endocrine. And the job of the exocrine is digestive enzymes primarily. All right, here's a picture here of one of these acinar areas here, the exocrine part. And here it is right here. There's the duct. These are the acinar cells producing these zymogen granules. Now, zymogen is an inactive enzyme, ogen inactive. Okay? Just like we were talking about pepsinogen in one of the last videos. All right, so let's go further. Exocrine acinic clusters of secretory cells secrete pancreatic juice. Zymogen granules of the secretory cells contain digestive enzymes, but in an inactive form. They'll be activated. Okay, we go further. Pancreatic juice is alkaline, neutralizes chyme, and allows pancreatic secreted enzymes to work. Okay. The epithelial cells lining the pancreatic duct secrete electrolytes. So these cells here secrete electrolytes. Okay. The bicarb is made in the epithelial cells. Here's the epithelial cells. The bicarb is made in the epithelial. These are glandular cells, and these are lining ductal cells, lining epithelial ductal cells, in case you're trying to figure out what all this is. So these cells here make pancreatic juice, pancreatic enzymes. These cells here do more of the electrolyte aspects, primarily bicarb, these cells here. The bicarbonate is made in the epithelial cell. So let's talk about that. All right. So here's an epithelial cell, let's say. And what's going to happen is that this is the pancreatic duct right there. So they secrete this bicarb. All right, this pancreatic duct. So right here, what's going to happen? Here's a pancreatic acinar cell. Let's go back here. Here's a pancreatic acinar cell that primarily does the digestive enzymes, but it also assists in the production of what? Some of this bicarb. Okay. And the epithelial cells kind of secrete it. They kind of make it, and the epithelial cells kind of secrete it. So let's see how this is going to work. So here's the blood vessel. All right. Water gets in plus CO2. You know what that does? Form carbonic acid. The carbonic acid then goes into the pancreatic duct. Okay. The H plus goes into the blood capillary. So the bicarbonate is made in the epithelial cells. For every bicarbonate secreted, H plus is returned to the blood. Now, do you remember the alkaline tide we talked about coming from the parietal cells? Remember we said if there was quite a bit of amino acids, protein, then you'd make a lot of bicarb, and that blood draining from the stomach is heavily alkali, alkaline tide. Well, the blood coming from the pancreas is acidotic. So it kind of neutralizes so in other words, the pancreatic tide right here is acidic, and the stomach tide is more alkaline. So it kind of neutralizes it. Okay. So we go a little further. Pancreatic juice, acinar cells, let's go back here. Acinar cells produce enzyme-rich secretion. Amylase breaks down carbs, lipase lipids, nuclease DNA, RNA are secreted in an active form but require ions of bile to optimal activity. Protease is secreted in inactive form. So these are secreted in active because, see, proteases would break down proteins, which is what you're made of. Okay. So the zymogen granules contain let's say the protease, which are inactive. Protease activation in duodenum, because remember now the pancreas sends into the duodenum. We already talked about that. The pancreas sends into the duodenum. There's the pancreas. Protease enzymes that the pancreas produces is trypsinogen, procarboxypeptidase, and chymotrypsinogen. Okay? Trypsinogen. Trypsinogen is activated in here by an enzyme called enteropeptidase, but that's made by the brush border of the intestine. So and we're going to hit this one more time. So when the trypsinogen goes in, enteropeptidase produced by the cells here can convert that to trypsin. Convert that from trypsinogen, inactive, to trypsin. Okay. Two other enzymes are also made that are protease. Okay, procarboxypeptidase, inactive, and chymotrypsinogen. But once trypsin is made, trypsin acts as an enzyme to activate these two. That activate. So you have to sit down and really study that. You have to really sit down and kind of study it yourself. Okay, so here we go. So other words, I'll come over here. Right there. Let's just take that up a notch. Let's just do it one more time. Let's do it one more time. All right. So we'll come back here. Trypsinogen is secreted from the pancreas. There we go. That's trypsinogen. It's inactive. So here comes in. Trypsinogen is inactive. The epithelial cells, brush border, inside the intestine, let's say here's the duodenum, secrete enteropeptidase. It's on the membrane. It's bound to the membrane of these epithelial. That acts as an enzyme to convert trypsinogen to trypsin. Once trypsin is made, it can help make more trypsin because it acts as an enzyme on itself. But it also acts as an enzyme to activate two other enzymes produced from the pancreas, one called chymotrypsinogen and the other procarboxylic peptidase. Now, we'll talk later as to why you need all these different proteolytic enzymes. Say, why you need so many to break down a protein? Why not just have one? We'll talk on that. So for right now, just accept it. But the trypsin once formed makes you become active and you become active. So trypsin is the pivotal thing to get active. Okay. All right. So we go a little further. Now we're talking about the gallbladder, regulation of secretion. The gallbladder contracts, and I brought this up due to something called cholecystokinin. Cholecystokinin. Okay. Let's take this back down some. Okay. So chyme entering, let's just do step by step. Let's do step. Chyme entering duodenum causes release of CCK and secretin from the duodenal mucosa. Okay, CCK and secretin. So before we do that, let's come here and talk about secretin. Secretin is secreted from the cells of the crypts of the upper small intestine. It's a polypeptide. The main function of secretin is secretion of bicarbonate and other electrolytes from the pancreas. So secretin stimulates the pancreas to release that bicarb we've been talking about. It inhibits gastric secretion, so secretin slows down the stomach to allow the small intestine to work on stuff longer. It maintains a proper pH, because remember, the stuff coming in the stomach is acidic, and the bicarb that's being secreted from the pancreas kind of neutralizes it. Okay, so its main function is secretion of bicarb and other electrolytes. We just said that's what the pancreas did. And okay, all right. So we go further. Then we brought up this CCK. Let's see what we got. So here again is secretin produced by the duodenum enteroendocrine cells, crypts of Leeuwenhoek, stimulated by arrival of acidic chyme in the duodenum. Function stimulates bicarb secretion, inhibits gastric secretion, slows the stomach down, and so forth that you can see. All right, let's go to CCK. Cholecystokinin, that's what CCK stands for. Cholecystokinin is secreted from endocrine cells that are concentrated in the duodenum, but found throughout the length of the small intestine. The postprandial, when you say postprandial, that's a fancy word for after eating, of CCK is the key in the activation of intestinal feedback of gastrointestinal. It stimulates the pancreas and gallbladder. And I want you to pay careful attention that it stimulates that gallbladder to contract. Primarily because of certain substances that are in there, fats and proteins, primarily fats. Okay, so that's the key. You can look at it, but that's really what I want to hit. So we go back. So gall is stimulated by CCK primarily, vagus a little bit, causes hepatopancreatic duct to relax so you can let the juice in. Bile secretion is stimulated by bile salts in the intrahepatic circulation. We already talked about bile salts. Okay, I already talked about bile salts. In case you don't remember, that in red are those are the bile salts. That's what really does the emulsification. The more bile, secretin also in HCl helps. Okay, so bile and pancreas are regulated by same factors, CCK, secretin, and the vagus, but the vagus not much. Okay, we already talked about this, CCK. So we're going to come back here right there and start on this one more time right there. Okay.